Assalamu alaikum wa rahmatullahi wa barakatuh. Today in this video I'm going to show you how to do seed germination test at home using seeds that are available at home from fruits and also from cereals. And by doing seed germination you will be able to calculate the germination percentage as well as the rate of germination. So let's get started and do our germination test. Okay, so this is what you need. First, an apple with hopefully seeds inside. This is sweet lime. In this case, you can use any kind of lime, Omani lime, or any citrus like oranges or mandarin, anything with seeds inside of it. Also get yourself dry seeds. What I have here is wheat and lentil, but you can use any kind of legumes like chickpeas, beans, peas. You could also use barley or corn or any kind of dry seeds. Okay, so the first tip is to extract the seeds from this apple. You know, apple is a temperate fruit. That means it grows in cold climates. So the seeds will not grow right under normal conditions when the temperature is normal, like 25 degrees. They will have to be subjected to treatment, and that treatment called stratification. Basically, you extract the seeds, put them in grown media. In this case, we'll not put them in grown media. We'll have them in tissue paper, basically, and that should be okay until they start to germinate. They should start to germinate in a few weeks, but you have to keep them in the refrigerator. So this apple doesn't seem to have too many good seeds, but that's what we have. We are going to do with what we have. So this is what we have here. Remember what I told you about monocots and dicots, and you can see here that these seeds are hosted in 5 carpels, 1, 2, 3, 4, 5. Remember, apple is also a dicot crop. So this is what I have. What you need to do once you extract the seeds, you place those in tissue paper. You need a tissue paper like this, okay? And then you just put the seeds inside the tissue paper. This is all the seeds I have. I'm not sure how many of these will germinate, but let's see. Once you have these seeds in the tissue paper, okay, then you basically wet them with water. So I'm going to wet these seeds so they are all ready for germination. This is require something called stratification. Remember, to break the dormancy, there are certain treatments for these seeds. One of those treatments is stratification, and this is used to break the seed dormancy due to thermal dormancy or cold temperature. Here is my label. You always have to have your label in. You can also have the label in this tray. So I'm going to place these in a bag and put this in the refrigerator. You don't need to put it in the freezer. The refrigerator will do fine. Make sure that you keep them wet until they start to germinate. My second sample is the sweet lime. In Oman it is called sfarjal. Okay, so I'm also going to extract the seeds from this citrus fruit, and I hope there are seeds there. Okay, there are a few seeds, which is good news for me. Here is a few seeds that are here. Okay, I don't see so many, but they should do. One thing you need to know about citrus, or this is sweet lime, that it is actually tropical and subtropical. This is subtropical, so it's not temperate like the apple, and therefore it doesn't go through dormancy, so it doesn't really require any special treatment to germinate. All you have to do is just extract the seeds and allow it to grow. Just place them in tissue paper under room temperature, make sure they are wet, and then monitor the growth. Another thing you need to understand about most of the citrus species and their cultivar is they are polyembryonic. Polyembryonic means each of these seeds will produce more than one seedling. So we don't know how many now, but we'll be able to see that once we have the germination started. And some of these seeds will grow more than one plant. One of them will be zygotic, as you know, and the rest will be nuclear embryos. Okay, mitotic embryos. And so the mitotic embryos will be prototype, identical to the mother plant. So the fruits will be just like this one, identical to it, but the zygotic embryo is not going to be, it's going to be different, and actually it looks different. So we'll be able to see that from this experiment. Now let's put these seeds, okay, also in a tissue paper. So I have my tissue paper, my tray here. You get your tray, you put your tissue paper, okay. Here is the tissue paper. Obviously you have all these. You can use oranges and you can use mandarins, as I mentioned before. You can use Omani lime too. So this is going to be much similar than the first one. So here we have 5 seeds, okay, and so we'll be able to monitor these 5 seeds and see how they do in terms of germination. Hopefully in a few days I'll be able to show you if they germinated or not. Now my tissue paper is wet, so it's good to just spread them apart. Here they are, 5 seeds of sweet lime. Then I have another tissue paper on top of this one, okay, and then this one doesn't have to go to the refrigerator, unlike the apple, because these are, you know, tropical or subtropical, and so they don't go through dormancy. Again, the same thing, don't forget your label, and then don't forget to keep it in a plastic bag so they don't dry out so fast, and keep watering these because these will be at room temperature. Okay, so this is another set of seeds. These are lentils, and as you can see, these are 20 seeds. And then what I did is I just had a tissue paper underneath in the tray, and then had my seeds there. And I'll just do the same like I did before, which is basically have a tissue paper also on top of these seeds, and then wet them. So these are 20 seeds of lentil. Lentil is legume. You can use any kind of legumes in this case, peas or beans. And then you need to water them regularly. Make sure you don't forget the label. You could also have the labels outside in the plastic bag itself. It doesn't have to be inside, because I keep it for display. I'll put it inside. You can also keep these seeds at room temperature and water it frequently, and they should germinate in a few days. And my last set of seeds are these wheat seeds, and I use what we call habharis. This is basically wheat seeds that we use for cooking. You can find it in any store. And then you do the same thing. You just cover them up and down, top and bottom, with tissue paper. And then all you have to do is just wet the tissue paper here, and that you are going to do a few days until they start germinating. Don't let them dry, because they will lose viability. Now the question is, how old or new? You decide how old or new your seeds are. If they are fresh, you could probably use the fresh seeds and also last year's crop, for example. You can also do the same thing with fresh seeds, like from apples or citrus. Extract some and leave them for 1 week, for example, to dry, and then also get a new one, fresh seeds, and experiment with that. I will leave that up to you to do whatever you think is important, but these are all the same steps. Basically, what I have here is the seeds in a wet paper and wet tissue papers, basically in a tray. You can find these trays in a store near you. It's not difficult, it's not complicated. And then all you have to do, just keep them wet, keep these under room temperature, and then water them. Don't let them dry. And then monitor the germination on maybe once a day or once every 2 days, and you'll be able to see the germination rate, which is basically the number of seeds that germinated over time, and the germination percentage, which is basically the number of seeds that have germinated at the end of your experiment compared to the number of seeds that you place in the tray. For example, if I had 20, like now, in my tray, and 10 of them germinated, that would be 50% germination rate. So this is basically it. We are done. Or now I have to do is just keep watering these seeds and take the data on their germination. So we'll see you in another video once this experiment is completed. Good luck, and wishing you all the best. Goodbye.